Hey everyone, thanks for joining, and thanks to Djuna von Maydell, who has agreed to give us a tutorial on identifying subgroups in biomedical datasets using a data attribution method. Djuna is a PhD candidate in the Brain and Cognitive Sciences PhD program here at MIT in the Thai lab, and we're very excited to hear about some methods she's been developing throughout her PhD. Thank you.
Thank you, Greta. So, yeah, so a lot of unfamiliar faces. So I don't quite know kind of what people's backgrounds are and what your research is in particularly, but I'm actually hoping that kind of walking you through this method, which actually, Greta, I didn't develop, I'm just applying, can be perhaps useful for really whatever you are working on or whatever kind of data you're working on. But because I am on the molecular side, the examples I'm using are molecular. So hopefully that's interesting to some of you as well. So really, I came into this topic through a biological problem that I've been working on during my studies here. And particularly, I'm interested in understanding whether there's evidence for different molecular subgroups in diseases like Alzheimer's disease, which are very complex diseases. And as inspiration, probably most of you know that the cancer field has made a ton of progress in precision medicine. So really understanding that in cancer, even a cancer type like breast cancer can have a lot of different subtypes within that cancer that actually use very different mechanisms to evade the immune system or to grow abnormally. And just as one example, for example, you can have metastatic, sorry guys, so the Zoom died for a second. Right, but so we know from this line of research that there are different subtypes of cancer whose features, so genetic signatures that differentiate these subtypes from the healthy epithelial tissue from which they derive. Sorry, guys, for the interruption. So I will be quick with the introduction and kind of just pointing out that subtypes that use different molecular mechanisms exist in biomedical realm. But when we look at Alzheimer's disease, identifying subtypes of the disease that actually use different molecular mechanisms to develop the pathology and the cognitive decline that we associate with the disease hasn't really been successful yet. And that's evidenced by the fact that we don't have any precision treatments for the disease, let alone really any treatments at all that work well. And many previous studies in this field that have tried to do some kind of subtyping analysis have really approached the problem by trying to select relevant molecular features from patient data, either kind of hand-selected or using linear models, and to cluster on those relevant features. And so that begs kind of the initial question, what are the most relevant features that we want to be using to try and discover whether subtypes even exist in this disease? And secondly, how can we actually evaluate that any of the clusters we identify are meaningful to the disease? So you can cluster on pretty much anything and identify subclusters, but how do we actually test in a principled way whether they are meaningful to the disease and the disease mechanism? So these are kind of two questions I want to get at. And I think that this question overall kind of goes hand in hand with something that in the molecular sciences we perhaps struggle with to a certain extent. But there's a flip side that is positive. So in neurodegenerative disease especially, we just don't have, in comparison, if you think of like an imaging data set in computer science, we don't have nearly the number of samples, right, for which a lot of these methods are developed. And that's because we get brain tissue from patients, right? So the number of samples over the years doesn't increase like in any exponential or any crazy way. But what is increasing is the amount of compute, right? We have better and better GPUs that we can use to analyze these data. So in this kind of regime, we have more compute than we have samples. But what we also have is more and more single measurements from every patient. We're getting, single cell measurements where we have 20,000 genes per cell, maybe 1,000 cells per patient from a single brain. So we're kind of getting all this additional information about individual patients. So we have this scenario where we have a lot of compute, not that many samples, and a ton of features to be digging through. And so we're in this, the real struggle is kind of finding that needle in the haystack. Like, how do we actually identify relevant biological features that are associated with the disease that can reveal different subtypes of the disease and so forth. And so in kind of getting interested in this problem, I really became aware of this method called data models that was a couple of years ago now, published by the Madry lab at MIT. So that's a machine learning lab over in ECS. And, they don't care or work at all on biological applications, but I think this is what makes it particularly interesting. And I'll really be describing my intuitive understanding of the method and kind of how to use it in a really practical way. But I want to emphasize that I didn't develop this method, so I don't want to overstate my expertise in it. But I'm hoping that you can get out of this the basic workflow for actually implementing data models, and then get a little bit of a sense for how we might use this method on our biological data, whether you're working on molecular data or all kinds of different levels in the human brain. So that's the goal. So briefly in the beginning, I'll introduce what this data models thing actually is. And we'll explore with you what the outputs look like of data models. And that's where we'll go to the Google Colab notebook. And I think I have some fun examples for that. And then I'll tell you a little bit about my own research and application of that, which hopefully will give you an idea of how we can leverage this method. Introduce more concretely the workflow. How can you train? This method relies on training thousands and thousands of classifiers. So how can you do that in an efficient way? And yeah, time for discussion. So if you want to interrupt at any point, just feel free to, if you have any ideas or points I'm missing. So briefly, I want to touch on the fact that previous studies in this, I'll call it like molecular Alzheimer's field, have attempted to identify important features that correlate with Alzheimer's disease without handcrafting features or using linear models. For example, by using Shapley values, where you're basically assessing the impact of removing a feature like a gene or so on model predictions during the inference phase, right? And there are just some examples here of biomedical and molecular data sets that have used this and found some interesting biomarkers and so forth. But what I think is less frequently explored in my subfield is actually the influence of individual data points, so samples, on model predictions. So we often think about features, but I think less frequently we think about how the samples actually influence each other, at least in this domain. So that's kind of where we're going with this. So how can we actually think about the influence of one data point, like a human patient in disease, on another data point in our data set? And kind of perhaps an intuitive way to think about it is this empirical influence estimation, which basically says, let's train a classifier on our data set, right, that is going to classify our held out test sample on whether it is part of the control healthy group or the Alzheimer's disease group. And then we're going to train lots of those models on our training data set, other Alzheimer's and healthy patients. And then we're going to do that all over again, train a bunch of models, but take out one training sample. And then we'll see whether we see a significant difference between the distributions, right? How confident and correct was the model on that held out sample when I keep that training sample in versus when I take it out. So that's kind of this intuitive, I think, understanding about in this way, we can measure whether this training sample had an influence on the model's learning and ability to predict the disease label right, on the held out sample. But I think you can quickly see that this becomes kind of computationally infeasible at scale because it involves training many, many models with all your training samples, many, many models without, I mean, maybe 1,000 or whatever you want to try. And then probably you will want to do this for every single training sample in your data set to get essentially an influence of every sample in your data set and every held out test sample. And also this approach kind of breaks down as soon as you have a very close duplicate. So as soon as you have groups of data points, which is what I'm getting at with this subgroup idea, right? That we have whole subsets of our training data or of our Alzheimer's population, let's say, that have similar mechanisms. So for which the model might learn similar strategies. If I hold out a single patient from the training process, the model will still learn those strategies, right? Those patterns from the samples still left. So this is where the data models method becomes very useful, because essentially what the data models framework allows us to do is make accurate predictions about sample influences. So what I was just talking about with fewer models. And instead of considering every single data point alone, it helps us capture population effects. And that becomes really important when we're thinking about clustering or subgrouping our data. And there's the paper on this is really great. So I really, if you're interested, encourage you to read it. It's super interesting with a lot of experiments. So I'll go through first just really an overview of what data models actually is. And so the Maydell Lab had a nice little example on their blog post for this. And really what we're trying to do with data models is get a system, a data model for a given target example, a held out test patient or image or whatever we're doing. And we want this data model to be able to tell us if we had trained our model on a subset of the training set, you know, these particular samples, how would that model have performed on our held out sample? So this idea allows us to make counterfactual predictions about what would have happened if, right? And we can leverage that for, as I'll explain later, to start and really tackle this idea of subtyping. So how does the Maydell Lab achieve this? In a kind of basic sense, what we're doing is we have our data set, right? Here, all the black dots. Those are our training samples in whatever domain you're interested in. What we do, what the data models process is we select a random subset of these training samples, shown here in the colored circles, of a given size, and I'll talk about that size and why it's important later. Then we train a model on each subset. So in this case, I'd have 3 models, and we measure the performance of each model on your test set, right? So we don't need to train a model separately for every test image. We can just evaluate it on the whole test set. And the intuition is that a model that is trained on a set of points and then evaluated on a test set that has a similar distribution, right, is in this circle or has a similar pattern to it, then that model will perform better than when it's evaluated on a test sample up here that is not part of that group. This is, of course, assuming, or maybe not assuming, but a way for us to actually test whether there are these different distributions in our data. Because if the models, or if all these training points had very similar patterns to be learned, then it wouldn't matter so much what subsample you take. And that's where I think it becomes really exciting for the biomedical application, because short of a clinical trial, right, where you take your patients and say, you're this subtype, that subtype, and I'm going to test that with a particular medication. And if you respond, then that's truly a subtype of the disease. I think this is actually a neat way to test this intuition without having kind of access to that. But we'll go into that more. Okay, so I briefly want to talk about this role of the subsampling fraction alpha. So I mentioned just now that you'll be subsampling this for every model you train, right? And so alpha is really the fraction of training points that you're including in every model training instance. And as alpha goes to 1, right, as you start taking every single training point into every model, you won't have any variation to explain because you're training on the same data all the time. As alpha gets smaller and smaller, you start dropping out larger and larger groups of training data, and you start getting variation in your outputs of how all your models are performing on a given held out test sample. And so there are trade-offs to these alphas. So you need to think about what alpha you're going to choose and perhaps try a number of different alphas, because a smaller alpha intuitively gives a given model less data to train on. So that model probably will be less stable or have less stable training and just higher noise, right? From initiating the model, from data augmentations or whatever you're doing. And a smaller alpha probably will also identify simpler patterns in your data because there's less to train on. But potentially you'll get more signal because you could be dropping out entire, in my example, disease subgroups. A larger alpha on the flip side would give you less noise, potentially more complex patterns, and less variance in the outcome. So that's kind of broadly, I think, the intuition. But there's some interesting aspects to this that I think we'll touch on in a little bit as well. Another important thing to note is that what is this variance in the outcome that I'm talking about? So when we are training all of these several thousands of models, for every model we're subsampling according to our alpha, some of the training set, we're training that model, then we're evaluating that model on every sample in our test set. And the variance in the outcome is really, is the model correct in its prediction, but also how confident is it in its prediction? So you can think about that as how far is a test sample from the decision boundary that this model has generated. And that's important because we're fitting on a continuous variable, and we have these nuances. Even if dropping out a certain group of training samples makes a model still predict correctly, it might be much less confident. And so we want to kind of get that entire spectrum.
Just to help me understand,
Yeah.
the intuition. So in the Alzheimer data example, would the decision be that this is from Alzheimer patient?
Exactly.
Okay.
Yeah, exactly. So that's the example I'm dealing with, but I want to make it clear that this is would apply to any classification problem, and the example is on the CIFAR data set. So what does this look like in practice? I like to look at the matrices, and actually I think this will give a really good introduction to how to actually do this. So really what we end up with after we've trained all of our models on, let's say, a classification task, differentiating healthy and Alzheimer's brains, we end up with a training mask matrix, and that has M model rows. So every row is a separate independent model you've trained, and the columns indicate which of the training samples were part of the training set for that model and which weren't. Next, we have our margins matrix. So again, the margins indicate how confident the model is, how far the margin to that decision boundary is. And again, we have our models in the rows, or however you want to do it. But this, we have our test samples. So whatever the models never saw, and essentially our confidence is for every single model on every single test, held out test sample. I want to just note that you can expand your test size by also including training samples that weren't included in a given model, right? But just for when we do the CIFAR example, it's so easy because they're already split, but that's just something to keep in mind, especially for biological data where we just don't have so much of it. And then what is a data model, essentially, this data model is some mapping, some function that takes as input for any given individual in the column, whether or not that individual was present in the training set of that particular model, and then tries to predict the confidence on a given held out sample. So what we're doing in essence is we're taking our training mass matrix, we're trying to find some transformation that will explain or predict the variance in the model confidences. So this, is that kind of making sense? So we're actually learning a transformation for every single test sample. And actually, just to go here, it's clear from the Madry paper that this transformation can just be a linear model. And that's what makes this whole thing so powerful. But you can also try other models, but the linear model is what gives us the intuition, essentially, that every weight in our linear model is telling us how much a given training sample is influencing the prediction on the held out test sample. So a large positive weight would indicate that a given training sample is having a positive influence, making the model correct and confident on a held out sample, and a large negative weight would have the opposite. And then you can, take a combination of those points, of those coefficients, and predict how a model would perform on a held out test sample for a given set of training samples. I hope it'll become a little more clear, but actually, once you start kind of thinking about it and going through the workflow, it's a very simple, intuitive way of thinking about it. Yeah.
Fill out the data points that have small margins, or you still want to keep them? Or sorry, not small margins, small coefficients.
Yeah, so that's the next really good point, which is when we fit this linear model to explain the variance and model performance for a given held out test sample based on the training set, we probably don't have enough compute to train as many models as we would like, that we would have enough samples. In this case, the models are the samples. That we would have enough models to fit this linear mapping without any kind of regularization. So in this case, in the paper, they show also that they regularize with L1, which is exactly, or getting to, I think, your point, L1 regularization tries to have as many entries be 0 as possible. And there, the intuition is that you would think that for a given test point, there are only actually a handful of training points that influence that test point. Versus L2 regularization would try and spread the influence over more training points. Yes. Yeah.
Is it G theta? Like, is that like a row from the margin matrix, or?
Yeah, sorry. I maybe didn't make this clear. It's just trying to essentially take that cartoon image I gave before, saying that this G theta, which is that linear mapping for a given test.
One mask array.
From one single model.
For one single model.
It takes as input and then predicts, if we had trained that model, how would that model perform on the held out sample? You could then take another row, try it for a different model, try to predict a different value in your margins.
So you mean if the mask vector for a specific model is determined, the margin vector of that model is also determined, which furthermore determine the confidence of the model?
I'm not sure I totally understand the question. Maybe let me try. So what you have to start with is your training mask matrix, where every row is a model you've trained, and every column is just telling you whether or not that training sample was in that model. And then you have one vector, which belongs to one of your test samples. And that vector shows you how the model the models that are in the rows here performed on that held out sample. So then you're trying to explain the variation in that held out confidence for that one sample as a linear combination of what training points were in a given model.
Oh, I see. So the marginal, so the margin matrix is for one single.
Yeah, I should have made that clear. So when you're fitting one data model, you're taking one column of the margin matrix. And you could ignore all the other samples. You could just do this for one held out sample. And then you would try to predict the model confidences in that column from the training mask and the weights that you're learning.
In a weight is fit.
Yeah, so you fit that. So essentially you split your training mask along the model axis, right? You split it into another train and a test set. Then you fit on the train set. You try to fit a linear model that is going to predict your margins in that column based on your training mask. Then you evaluate it on the models you held out, so maybe the bottom rows of that training matrix. And then you do that again for the next column and your margins. It takes a little bit to wrap your head around. And I think I'll also share the code so you can like look through it. But I want to keep going also to make sure we get some of the intuitions.
Ask a quick question.
Yeah.
For example, if for the first row, if second sample is 0, which means for the margin matrix, the sample 2 on the first row need to be smaller to have a better prediction.
No, not necessarily. All it means in the training mask is that that particular sample wasn't in the training set for that model. So then you're just trying to explain the variation in your column. So in model confidences, right, you have a single held out sample. It has a lot of confidence. It has a distribution of confidence scores associated with it, which come from evaluating every model on that sample. And we want to explain that variance in terms of what training samples were in a given model. It'll hopefully become a little bit clearer as we go along. But I do want to make sure we get time to actually explore what the data models do and why they're cool. So very briefly, I want to highlight a couple of results from the paper, and this might also help clarify some of this. So what they show in their paper is that they can very accurately predict how a model is going to perform on a given held out sample just based on knowing the subset of training points that were used. So essentially, they show that these data models are very predictive of model confidences on your held out samples. So even if you kind of leave to aside for a second the matrices and exactly what we're doing, this means that we do have this kind of black box, right, that can take as input a target example X, like your held out Alzheimer's patient, and then say, if we had trained a model, that's what we're doing here. We're doing that inference, right? If we had trained a model on this patient, that patient, and that patient, how would this model have performed on our held out patient? And that becomes very powerful when you start looking at what that can do. Yeah.
In this graph, what does true margin mean? Since it's a classifying.
Where do you see?
the number one accurately predictive model predictions in the graph.
Right. So the true margin, of course, to test this data model, they actually they train those models and see whether they can predict with their data models the actual outcome of training those models. Does that make sense?
Or I guess what we know for truth is, whether this is like, Alzheimer or healthy, or I guess the image example, whether this is a plain model and so on. So to me, it's like a binary thing, or like the
Right.
margin. Yeah.
The margin is continuous. Yeah, so that goes back to, we're not trying to, for every held out sample, we're evaluating the models that we trained, and our output of that evaluation isn't just yes or no, the model was wrong, but it's how wrong was the model. Yeah.
It's true margin if you take the global like sample, or is
No, so in this case,
I guess I'm just about the wording, true margin, right?
well, predicted margin is the confidence is predicted from our data model, right? So every point is a model that you're feeding through your linear function, and then predicting, if we had trained on those samples, how would I do on a held out sample? We can actually train that model and really evaluate how the model did. So that's our true margin.
So you mean like they actually train a model, use the mass matrix as input, and it can predict the predictability of a model.
Yes.
Just using that mass
Yeah.
matrix.
Yeah.
So
It's
a
predicting
model that
predictions.
predicting prediction.
Yes.
Okay.
Yeah, that's the intuition. It's a model that is predicting the prediction.
And is margin how wrong it is? I thought margin is like how the confidence, how robust the margin is.
Yeah, I think it's both. My understanding is the margin is telling us not only on what side of the boundary that this model is learning that sample falls, but how far.
Yeah.
Like,
Is farther the better?
yeah, farther would be right, like more confident in the prediction, but it could be on the wrong side, which is negative then. Yeah. So I hope that's becoming clear. So it is actually, yes, it is predicting predictions. So it's kind of meta. So what can this data models framework actually do for us? So in the paper, they do this thing on the CIFAR data set. They train a classifier to predict all the 10 classes. And then they use data models to predict predictions. And they start looking at the weights in this linear function that is predicting these predictions. So again, these weights are telling us how much a given training sample is influencing a held out test sample. And we'll go into this in detail, so just kind of appreciate maybe the high level here. For example, their held out sample here is a plane. They see that when they look at the top influencing points in the training set, these are all very similar types of planes. When they look at the influencing points that really hurt the model, so make the model confident but in a negative way, right? It's getting it wrong, but it's confident. Then these points are, you can see why it's confusing the model, right? Because they're not planes, they're animals, but on a similar background. And this can start giving you intuition about what features the model is actually using to make its predictions. Then what is very cool about the data model's embedding, because in the end, what you have is you have a matrix of where the rows are your training samples and the columns are your held out test samples. And every value is giving you that coefficient. It's giving you the influence of that training sample on the held out test sample. So this embedding, this data model's embedding, if you, as they do here, project it onto your first few principal components, you start seeing really intuitive features that differentiate these samples. So as an example here, the principal component number 1 is clearly differentiating between horses and dogs, but in very similar positions, right? So it's elucidating the types of features that the model is paying attention to in making its predictions. So what we can do on this data models embedding is start to cluster in the space and see whether we can identify meaningful subgroups, right? Groups of data points that are influenced similarly or influencing similar data points in a way that if a model is trained on one subgroup, it performs worse on other data points outside of that subgroup. So it I think raises this really interesting or gives us this interesting and intuitive tool to start thinking about subgroups in our biological data. Because if we can identify these subgroups in our data, which of course wouldn't be labeled, we wouldn't know that these are red boats and white horses. But if we can identify subgroups and then use the information from the data models embeddings to guide counterfactual predictions, like if I train only on the red boats now, I should perform badly on whatever the white boats or the white horses, right? And vice versa. And so we can make these tests, do these tests in our data to really try and elucidate whether the subgroups we're finding are meaningful. So I know that, oh, sorry, yeah, Fernando.
In the image space, it's kind of, sometimes it's a little obvious, like what features are clustering. Is, do you find something similar in the biological data? You're probably gonna have to get into it.
I'll get into it a little bit. It's definitely more difficult, but I hope that the tutorial will give a little bit of intuition in that. But the short answer is yes and no. It's harder to know, but I think so. Okay, so I thought that kind of in starting to get at this question of using data models to identify meaningful subgroups in biological data, I actually thought it would be cool for the tutorial to kind of do an experiment where we do something slightly different than the paper. So instead of training a classifier on CIFAR that classifies or learns these 10 classes, we trained a classifier on CIFAR that just learns inanimate versus animate objects, which approximately splits the data set in half. As a way to explore whether we can recover some of the subgroups like cars and airplanes and so forth, and perhaps even subgroups that we know exist using this method, because when we apply it to our biological data, we obviously don't know whether we have no ground truth labeling of our subgroups. So I think this is a good example. But I did start training the models this weekend. So I have about 60,000 models. So again, these would be all my rows, right? Which is enough for us to get some interesting signal, but it certainly hasn't converged yet. Like I'm still need to train quite a few more, maybe 100, 150,000. And also I've used an alpha of 0.1. So at every model training step, I'm sampling 10% of the training data. And that is also something that would need to be explored, what alpha is best for recovering these subgroups. So I don't know what the best way to do this is. I don't know how much time we have left.
Still like 40 minutes.
Okay. So why don't you guys, if you have a laptop like with you, open the collab and I'll open it here as well. So yeah, I tried to make, so if you have it open, I've written out some things and made it so that you can walk through it. So maybe just take like a couple of minutes to just run some of the cells in the notebook. And yeah, if you have questions, I can walk around, but we'll go through it in a moment. But just to start playing with some of the outputs, maybe I'll talk a little bit about it while you're looking at it. Does everyone have the link? All good. I might start kind of walking through it a little bit. So you can see, I hope. Okay, so in this experiment, as I said, we trained 60,000 models, classifiers that are learning to classify inanimate versus animate images in the CIFAR 10 data set. To speed up training, because this is a relatively simple task, I used half the training data set. And this is a comparatively simple task compared to trying to predict every single class. But what it does is it allows us to investigate model behavior because we have some information on these subclasses, these labels of airplane, dog, etc., that the model has not seen explicitly. And I just have a note here about why are we using this data set, not a biomedical data set. Again, just to illustrate with known subclasses, what this data model approach can do for us in terms of revealing some of those subclasses. So what we're doing here is just loading the results from the data model training. So what we have are these this data model's data set with the weights and the biases. So these are the weights from the linear model we fit, which tell us about the influence of a training data point on the held out test data point. And the bias is just the bias of how well that a model is doing on that held out patient. And then we load the CIFAR 10 data set. And so what I first wanted you to do is just have a look at the lower dimensional projection of the data model embedding. So when we have this matrix, which is telling us how our training samples influence our test samples in model training, what happens if we just project that onto the first components? What does that look like? So the first thing I want you to kind of make note of is that when we project from the data model's embedding, then the variance we get is much more spread out across our principal components than if we project, let's say, without having done data models, just from the pixels, like sample the same number of pixels from our images, where you can see, right, the skew much earlier on. And the intuition here is that data model embeddings seem to spread out the variance over a much higher number of dimensions, leading to much kind of higher effective dimensionality. And this becomes really interesting when we actually look at what these dimensions are. Okay, so what we do here is we project the full test data set onto some principal components. So let's look at principal component 1 to start with. Wonder if we can make this. And you can see that this principal component seems to be separating cars and animals, right? And this is the first principal component. It's the largest variance is represented here. And this isn't a huge surprise because we are training on inanimate versus animate objects or images in our data set. And you can explore some other principal components. So for example, PC2, this one is quite interesting because it's revealing, it's essentially a feature that is separating these red cars and trucks from planes and things on blue backgrounds. And this is starting to give us separation of our known subclasses, even though the model was never trained on the label airplane or truck, right? It was only trained on inanimate versus animate. But by subsampling training points and learning, allowing models to learn different patterns to solve this problem of classifying inanimate versus animate images, we're able to reveal some of the underlying structure in the data set. Yeah.
So I remember originally the training, you have the rows, those are number of models, and then columns, number of samples, right? When you say you load the models you trained, do you mean all the models they have the same structure, they just use different symbols, symbol.
Yeah, exactly. They're using different trained samples, but they're of the same family. Yeah.
You mean the model structure is the same,
Yeah,
just the input is different?
yeah, just the training samples. Yeah, exactly. Okay, and then we can project the, then we can look, subset our data and only look at, let's say, the animate objects or the images and project those. And then we also see some interesting things where, for example, PC3 is, to a certain extent, as I said, it's not enough models trained yet, separating these animals with 4 legs on a green background from, sorry, this is not the right example yet. Here is the example where we're looking only at inanimate objects. And we can see that even within this class, which the model was explicitly trained on, it's clearly dealing with different subsets of the data differently, and learning different patterns for different subgroups of the data to solve this problem of classifying inanimate versus animate samples. Is that intuition kind of making sense? Here's just another example of this, where you can also think of this as all these examples here, all these images are having a positive influence or are influenced by similar images, right? Other red trucks. And these examples here are influenced in a similar manner. So when you include in the model other red trucks to train on, then that model will perform better on held out samples that are also red trucks, because it's learning some kind of pattern that relates to some quality of this subclass. And yeah, there's some examples here that you can play through. But I wanted to also give you this example, which is another thing we can do is look at, for a given test image, held out test image, what is the top influencing train image? That if you include that train image in the model, it will improve the model's confidence and accuracy. And so for an example, you see this cat, I guess. This image is classified correctly with higher confidence by a model that is trained on a very similar image, right? And the same for here, the plane and the plane. Again, these are things that the model was not explicitly told labels about planes and frogs and trucks, but it seems to be learning meaningful patterns. And when you conversely look at the top negative influences, so the images that if you include those in the training set, confuse the model, perhaps counterintuitively, these images are very similar to the model, the image that it gets confused on. And that can tell you a lot about what the model is, what kinds of patterns it's learning. So for example, for this test image, a white car on a red background, the image that confuses the model most is an image of an animal on a red background. So the model is learning something about this red background in its quest to differentiate inanimate and animate objects. So this is a very different field from what I was talking about in cancer and Alzheimer's. But if we bring it back and think about a cancer data set, breast cancer data set, where we know there are subtypes of cancers that have very different molecular signatures from healthy tissue, then you could imagine that a model trained in classifying control versus cancerous tissue would do much better on tissue of the same subtype, right? Like of a white car on a red background, than on a cancer that is of a different subtype. So this is where this connection is coming in. That seems very disparate, but it's actually like this method is very useful. There are other things you can do in terms of clustering. But one thing I want to point out before I kind of wrap up with the biological part is that if you have a look at this perhaps later, we can run a really simple experiment to understand how the data model's embeddings might be useful to downstream tasks. And what we do in this example is in the first instance, we try to just train a linear classifier on a random sample of pixels from the CIFAR data set. And then we see if we can actually predict a single subclass, like dog versus not dog, just with those pixels. And as you can imagine, it doesn't work super well. That's why we're not using linear models to do that. But in the second instance, we'll train a linear classifier on a subset of data model embeddings, and again, try to learn a single class, dog versus not dog, just from those embeddings. And the striking thing is that we can do super well on predicting a subclass from the embeddings, even though this data model matrix comes from training a bunch of models that never saw the label dog or not dog. The only thing it saw was animate or inanimate. And I'll show you the data here. But essentially, if you just take a random sample of pixels from your CIFAR images and try it to fit a linear model that will predict a subclass, you can see that as you increase the number of pixels on the x-axis, you actually start doing worse because having fewer pixels is regularizing. When you really increase the test size, so really decrease the number of samples you're training on, you do pretty badly for a classification task. But when we do this exact or similar thing on the data models embeddings, so here instead of pixels, every value is a number of features that we're training on, then you can actually see that our validation accuracy really increases as we increase the number of data models features we're training on. And that goes back to the PCA plot I was showing you, that each of these features has a lot of information. Like I showed you some of the higher principal components, and they're differentiating for us really interpretable things in the data. And the striking thing is that now you can actually train a linear classifier on a really small amount of your data, 10%, and this is only the test CIFAR data set, so effectively 1,000 samples, and get over 85% accuracy on your validation set. So what I'm trying to say here, and I've tried to summarize it here, is basically that we can use data models to learn things about our data, about the underlying structure of our data. And then we can actually use very simple models. So a linear classifier, the simplest thing you could think of, and obtain really good accuracy and generalizability on a really small amount of data, on a really small amount of labeled data, essentially. So what this means is that from the biology perspective, where it can sometimes be very difficult to access certain types of measurements from, let's say, a human brain. Maybe, wait, I'm going to go into this in the last part and with a slide, but I hope that even if not every aspect of this is clear yet, that it's given you a little bit of intuition as to what data models does.
I have this understanding, I don't know whether it's correct. So for example, in this case, you have data models trained on very simple task, animal versus no animal, right?
Mhm.
And then you generate embeddings, that kind of like you learn embedding,
Yeah.
data model embeddings from
Yeah.
here, and then you're gonna have a brand new data set.
Mhm.
I have a different task, but it's also like dogs versus not dog. Anyway, a different task.
Mhm.
And then you, instead of using the image in this new data set, you just use the embeddings to learn from data models. And then
Yeah.
the more embeddings, the more features. So the more
Mhm.
features basically what you get from data models. And then-
Exactly. Yeah.
So in this case, you use the features here, and then you don't need a large training data set. You only need, for example, 10% training
To
because
learn
Yeah,
something.
the test is 0.9.
Yeah,
Right. So
exactly.
a smaller
Yeah.
smaller training set, and then
It's
very
giving you.
simple linear classifier, and then you
Yeah.
can get a very good performance. So that's
Mhm.
the benefit from data models for your downstream.
Exactly. And so exactly. And so just to clarify, the data models features are your samples that are influencing your other samples. So instead of now taking a pixel value for each sample in your validation set, and then trying to learn a classifier to predict dog versus not dog, you're now taking this data model embedding, so how these other samples are influencing that sample to train that classifier. So it's, yeah, it's what, I think you got it with what you were saying. But yeah, just if you're interested in it, you know, have a look through the notebook, just play around with it a little bit. Yeah, just to,
But
guess.
it's
Oh, yeah.
for training data models, it sounds like you're training a lot.
Yeah, so exactly. So I'm gonna go into this now, hope a little bit briefly, so we're not running too much out of time. I'll very briefly touch on the biological part, motivation. And there's some notes here, but I'll share the slides so you can go through them if you want. So again, this biological motivation is, can we devise some way of testing whether in our human data sets, let's say genetic data or gene expression data from human brain of healthy individuals and people with a disease like Alzheimer's, can we somehow test this hypothesis that there are multiple paths to different subtypes of the disease? And you can perhaps intuit these paths a little bit as like different patterns that a classifier would have to learn, right, to differentiate the healthy from the diseased tissue, because whatever it learns on the blue group wouldn't generalize to the red group if these subgroups exist.
Would this be
But
the
you're
TCs?
exclusive enough.
I'm just thinking those different subgroups where you get kind of like similar to the PC, different PCs you get from data model. So each of those PCs, they represent one of the traits or factors.
Yeah, I mean, some feature that is differentiating perhaps a subset of these samples or these subgroups. But you would need to investigate what the features are actually telling us, because in the CIFAR data set, we can see pretty easily, but in a molecular data set, it will be harder to actually know.
And so I have another question. For example, in the collab you showed, it's all about images.
Yeah.
Right? So you have the task trained data models on images, and then you have a different task, but it's also image related.
Yeah.
And then, for example, I'm working with brain, but as my data, I have this kind of, I want to get subgroups like autism or ADHD, but I only have this limited number of data. And then if I train my data models here, so I just want to know
how to have like train and test.
Yeah,
or,
and then
yeah,
perform downstream tests.
Yeah.
I only have to,
no, perfect. That's actually exactly what I'm talking about here. So that's a really good question, because again, with the CIFAR data set, we have this separate train and test set. But as I mentioned before, you can try and expand a little bit, right, by using the samples you don't train on as your validation set when you're doing the data models. So whenever you train a model on a subset of your data, you then evaluate that model on the samples you didn't train on. So that's what I did here. So I have here only 450 human brain samples from healthy individuals and people who have a clinical diagnosis of Alzheimer's disease. And this is a pretty simple, it's an XGBoost model. And I haven't done a ton of tuning on this yet, but you can see that we can predict from this molecular single cell data that we have this Alzheimer's label with cross-validation. So that's something you could do. And then we run our many models. In this case, maybe I did 100,000 or so. I trained 100,000. And from that, we can start to estimate our data models that predict model performance on the held out samples. Then we take those weights from our data models. So again, the influences of our training samples that were not in a model, on the held out sample. And this is, for example, what these influences look like just within the Alzheimer's patients. So, for example, in your case, right, and a really important kind of part of applying this method on biological data is what label are you going to train on? Because you want that label to be meaningful enough to your task, right? Like we train on inanimate and animate on the CIFAR data set, because we think that the model will learn something meaningful for these different subgroups. But if you train on something random that has nothing to do with that, you won't get the signal that you're hoping for. So in your situation, I don't know exactly what the data set is, but I'm assuming you have some healthier and
Yeah, so it's brain data FMI. So I have healthy controls, and then others we have different developable mental like disorders. Sorry, and then so I was thinking, so for data models, maybe you want to train on a broader, like a larger task. So for example, control versus having disorders.
Yeah.
But then your downstream tasks,
exactly.
you want something smaller,
Yeah.
so you can use the features because they will
Exactly, yeah. And so it also depends on what question you're asking. Like in my case, I want to test the hypothesis that these subgroups exist. I don't have any I have some labels for a more nuanced diagnosis, but not as clear as in your case, for example, where they're much clearer, separate diagnoses.
but they also have the mobility. So
exactly. So this method would help you test whether there's kind of intrinsically evidence for these clinical diagnoses.
So, for example, if I don't have brain data, I only have the, just the clinical test. So it's a kind of behavior, not just questionnaires. I think this one should also work, right?
Yeah. And we can brainstorm more. It sounds super interesting. But I just want to show you that, like, we see structure in this data models matrix for our AD patients. So these guys, the yellow, ones influence each other more, influence model performance positively. And so there seem to be kind of subgroups in this data that when you train on them, they do better within than outside of. So you can see there's structure in just projecting on the first couple principal components. And now what I think is actually very cool, because is a counterfactual test that we can run with this data to start to really get at this question of whether the subgroups we're identifying, because then maybe we cluster with k-means or whatever on the embedding. But then we want to know, is it actually meaningful, what we're finding? And so what we can do, for example, here I've split the data along component 1 into 2 groups of Alzheimer's patients. And then I train, and essentially what that is saying is it's predicting that this group, if you train and evaluate within that group on tests, you split, of course, then you're going to perform well on that group because they're similar. But you're going to perform less well on that other group and vice versa. So you can test it. You can just train those models on, because again, data models is giving you a prediction of what combination, you're not actually having to train every single model to get this data models embedding. And then you can test this. So in this case, for example, I took all of the AD samples within a cluster. So either all of the ones on the higher or the lower end of that split. And I trained and evaluated within that cluster, or I trained but evaluated on the other cluster. And you can see that model performance really, it improves when we follow kind of the guidance of the data model embedding.
In the same cluster?
Yeah, and train and evaluate in the same cluster. And it really degrades when we train in one cluster but evaluate on the other. So again, suggesting that the model, it's kind of like the truck example, right? It's training on trucks and other images to differentiate animate and inanimate, but then you're evaluating on a different subtype.
Would this be like circular, so you train this and they generate clusters and then you train and validate within the same cluster?
Well, I think it depends on, I guess, what your ultimate goal is, right? Like maybe this is debatable, but in my mind, I'm trying to find splits of my Alzheimer's population that will have different features that distinguish them from healthy brains, like the cancer data set HER2 positive or HER2 negative, but possibly more complicated features than that. And I think actually a very good test for that is if I have a split in my data, let me train, and I want to test whether these are two different subpopulations. I train on one and evaluate on the other. And if my model doesn't generalize, right, then it hasn't learned something in that one that it's able to use on the other, is the logic.
Prediction. So like counterfactual prediction. I don't know this definition. Like,
well, I think is
evaluating on another data subset.
Is the idea that is that essentially what you were saying about making predictions about predictions.
On the fact, like, so
like if I had done this, then that would have happened,
Okay.
because I didn't actually train the model yet. It's just telling me, according to the data model embedding,
manually introducing the like how to say the like the balls of like so you actually generate a artificial boundary. Am I right?
yeah, so I use the data models to guide my split into subtypes. And then I ask, according to the data models framework, this subtype is more related to within itself than to that subtype. That's what the data models embedding is suggesting. But then that's kind of the counterfactual prediction we're making based on that embedding, is that if we train within that subtype, then what we learn for our task that is relevant, right? The Alzheimer's features that we learn are not generalizing to this other Alzheimer's population because they have different features that distinguish them from the healthy brain. But if there were no evidence of subtypes, then data models, probably we wouldn't see this structure in the data. But even if we did, we could run this test now, right? Which actually involves training these models, and the model should generalize if it's not true.
When you plot the right image, the right plot, do you use the embedding and projections as the input, or you use
No,
the original?
I go back to the
Okay.
original. Yeah. So yeah, that's a good question. So I guess we're kind of you guys can, yeah, absolutely head out. But if anyone wants to still hear a little bit, I'll just wrap up the last couple slides. So the concrete workflow is actually very simple. So you want to train a good model that is relevant to your task. You want a model that has a good signal to noise ratio, right? You don't want a model that every time you train it, it performs very differently on your validation test set, even if you didn't change the train set just because of the initialization or whatever. So that's the first mistake I made, is training a model that wasn't very good. And so my signal to noise ratio was very off. And that obviously makes it harder to explain in terms of the training data, which is what you want to do. The second important point is you want your model to perform well on the alpha that you pick. So if your alpha is low, so the number of training samples that you're training on is small, you want a model that is able to learn something from that small subset of your data. And you might need to play around with different alphas to find one that works in your situation. And also kind of be aware of the fact that if you're training on a small alpha, your model is likely to learn simpler patterns. So you saw on the small alpha in the CIFAR example, there was clearly a lot going on in terms of what is the background color of the image. So it's learning kind of simple strategies. But the trade-off of increasing your alpha too much is having perhaps not enough variance to explain. So you need to kind of play around with that. The second part is really spend quite a bit of time on getting a good accuracy on your on the alpha that you're using, because even a really small boost in accuracy could mean that the model switched from making kind of learning kind of silly patterns that might actually generalize okay across your data set, to learning a combination of less silly patterns, like different patterns for different subgroups. So you need to make sure basically that the model is learning things that you care about or are relevant to you. And some of that will become obvious by looking at the data model's embeddings, because you'll see what is, like in my example, I see that the model is using a lot of the background color. And that may or may not be useful to the subgroups I want to identify. Okay, the key to this is, of course, that you have to train a lot of models quickly. So I think the biggest thing I learned from the Madry lab is actually, before parallelizing and buying more compute, and maybe you guys know this, but I didn't, is really just try and focus on training a single model really fast and really reliably. And there's this blog post that I'll share if you guys want it or give me your emails, that just gives this really nice breakdown of like training a neural net. And first, doing the simplest thing you can think of, like really checking your code, making sure the data loader, nothing is messed up, and just building step by step on that. And for me, that was really helpful because I hadn't trained these models before, and it was helpful in like debugging. Okay, and then back to kind of training a lot of models fast. Data loading is often a bottleneck, especially if you have a really lightweight classifier. So in our case, if your alpha is really small, then the data loading part itself, because these models will train very fast, can be a bottleneck. So at the end of the slides, there's some resources. There's a library called FFCV, which is really speeds up the data loading part. Then once you have that, you've really profiled the data loading, you've made sure that your model is training fast. Then you can start training a lot of models in parallel, and you can use things like Ray, like these dynamic schedulers that will help you train maybe a couple of models on the same GPU. And that, because again, these models are small, and they can kind of interleave so that you don't have any compute that you're wasting, essentially. So I think the take home is, if any of you are actually considering doing this, I would be super happy to sit down with you and share my code and show you what I've learned from this. But the take home is that investing some time in the beginning to really get a good model, to really make sure your training is fast and efficient, will save you a lot of headache down the line. And I guess I really just wanted to end with this last kind of linking data models back to biological application. When we think about like all the biomedical data sets that we're having access to, we can think a little bit about cheap versus expensive measurements that we can take. So like an expensive measurement is you take a piece of a brain and do single cell sequencing on it. A cheaper measurement is you draw someone's blood and you measure some markers. So one idea would be, right, for breast cancer, for example, maybe you collect data points from breast cancer versus non-cancer patients that are cheap to collect, like blood work or DNA sequencing. You train a classifier on that data to predict cancer versus healthy states. You get your data model matrix, which tells you the influence of your trained samples on your test samples. or in your case, where we have less data, you could have all versus all. And then maybe for a few test samples, you determine the cancer subtype by actually assessing a more expensive feature, like something more invasive. Maybe you have a tumor biopsy or something. And then with these very few labels, because we saw in the practical part that you can, just from the data models, actually get away with very few labels to differentiate between a healthy and a particular subtype of your disease, then you could try and learn a classifier that will essentially help you label other data points with this subtype, like HER2 positive breast cancer, which is a much more expensive type of data point to get, or type of label to get. So I'll leave you with that. Maybe you can mull it over. And I've included some resources here. If you're interested in more information, like the data models blog post explains really intuitively the whole framework again. The paper is really great. It took me like quite a few reads to get my head around everything. The FFCV repository definitely use. The MadriLab provides a data models repository and also data that you can download to play around with from the CIFAR dataset. But I'd be super happy to share, if you're thinking more in terms of this binary subtyping, any of the code. So please let me know. Oh, no, sorry. Just if you're interested in these applications that I'm talking about, and want any help, then I can definitely help better on the code I've written. But I just meant like the CIFAR training that I have is that inanimate versus animate labels. And then there's another kind of advance to data models that is quite a bit more complicated to understand, but leads to even faster, slightly lower performance, but even faster. And with that, thank you so much for listening. And thank you to the MadriLab for both a lot of input on this and a lot of compute. And thank you, Fernanda and Greta, for letting me discuss this. And please feel free to email me if you want any of the code, or if you have ideas or want, to get some of the experts from the MadriLab to help you. Just let me know. Yeah, be happy to share.